Check sound check. Check one check. Come in, come in. Check one sound check. अब check है. थोड़ा सा नीचे कर लूँ उसको. Check one. Check one. A very good morning to all of you. Thank you for joining us 15th India Probiotic Symposium. Check one sound check. Check. A very good morning to all of you, and I welcome all of you to 15th India Probiotic Symposium. We will be beginning very soon. Till then, make yourself comfortable and thank you for your patience.
Check. Hello. Check. Hello.
For the ones who are not present in the hall but can listen to me, please join us here. We are about to begin.
Check. Hello.
I request all of you to settle down so that we can begin with today's first session. And once again, I welcome all of you to 15th India Probiotic Symposium. This is the day 2 we have gathered here for, and we're going to begin with the first session of the day. Good morning, everyone. Our esteemed chairpersons, speakers, delegates, senior international colleagues, and our young investigators for today. Now we will start our first session of the day on gut microbiota, probiotics, and interaction with host metabolism. May I invite Professor Bindu De, former Secretary TDB and retired advisor DBT, Government of India, Director Research and Development Cell RDC, Swami Rama Himalayan University, Dehradun, Uttarakhand, India, and Professor Pankaj Malhotra, Professor and Head Department of Clinical Hematology and Medical Oncology, Postgraduate Institute of Medical Education and Research, Chandigarh, India, to chair the session. Let's have a huge round of applause for our chairpersons. Thank you.
Good morning, friends. Welcome to the first session of the second day. We had very exciting talks and lot of leads on basic science front and also on the clinical trials. And then in the evening we had wonderful Bhangra dance. So you all are feeling energetic to share your thoughts for today. We have got 3 speakers for this session. The first speaker is Dr. Javed. The second speaker is Dr. Anil Kumar. And the third speaker is Dr. Tarini Shankar Ghosh. Dr. Javed, you are the first speaker. You'll get about half an hour. And I would request all the speakers to stick to the time because many of the dignitaries would leave, for their flights in between. About Dr. Agrewala, he is primarily, he is a PhD from SN Medical College, Agra University. He was former scientist and professor, and he's currently former chief scientist and professor in IIT Ropar, if I believe. Earlier, he was in IMTech, Chandigarh. His specialization is on immunology of infectious diseases and gut microbiome. He has got about 135 publications. He is the Shanti Swarup Bhatnagar awardee. National Bioscience Award has been conferred to her. He is having Fellowship of Medical Research Council UK, Bill and Melinda Gates Foundation Award for High Quality Research, then UP Science Award for Lifetime Achievement, etc. He's a fellow of INSA, IAC Bangalore, and Nasi Allahabad. Dr. Agrewala, please.
Thank you, Dr. Bindu De. And I'm grateful to the organizers, especially Professor N.K. Ganguly, Dr. Nija, Dr. Neenam Taneja. And today I'll be talking about the unveiling the impact of gut-mediated gut microbiota, the spices of the loss of dendritic cell tolerance. So currently I'm working at Indian Institute of Technology, Ropar. And we did this, most of the work was done at my previous institute, Institute of Microbial Technology. So first of all, I would like to thank my team, and the work today I'm going to present is mostly done by Hilal. And also it's done by Javed. So we have been listening a lot about gut microbiota since yesterday, that it can control many things, including it can control different type of diseases associated with whatever organ you wish to see over here. And of course, recently it's been also shown that it can modulate the immune system. And the vaccine efficacy can be modulated by gut microbiota as well, and as well resistance to infections. So recently there were two very interesting papers, and I've been allotted 30 minutes, but this clock is showing 15 minutes. So recently there were, in 2020, there were two very good papers, and these papers, they showed that eosinophils, they regulate the inflammatory response which is induced by adipose tissues. And ultimately, it can have impact on immunological fitness in old age. And another interesting thing, interesting study was done that when eosinophils from young, they were transferred into the aging, and it was observed that the aging was reversed. So everybody feels like they should have, they should remain young, even though they become old. So how this aging can be delayed or aging can be reversed so that you can start looking again young. So in this context, we observe that gut microbiota can impact eosinophils. So we thought that one of the important cell of immune system is dendritic cells. So it is these cells which can only cell that can activate naive T cells into effector cells, and they get ultimately differentiated into effector T cells and memory T cells. So these dendritic cells, they are quite diverse, since they can activate the immune system, they can activate naive T cells, as well as they have tolerogenic capacity as well, so that they maintain peripheral tolerance. The cells which have escaped thymic education, and these autoreactive T cells, teeny tiny population which escape thymic deletion, they remain inside a body, and these are the dendritic cells which ultimately are responsible for keeping the cells of the body, especially like T cells and B cells, into tolerogenic state. So to keep this in mind, we thought that what may be the impact of aging on the gut microbiota? Does it induces the dysbiosis of gut microbiota? And if it does induce, then what is the impact on dendritic cells? So in that context, we started characterizing dendritic cells from young animals as well as aged animals. And then we thought, if the young animal's gut is disrupted, dysbiosis, how do these young animals' dendritic cells, they behave, whether they have capacity to activate T cells, or they can induce tolerance or activation of T cells. Can their interaction leads to NRG, unresponsive in T cells, and then we also did analysis of gut microbiota and how does it changes with age, the microbiota stratification, and ultimately whether if there is any disappearance of a particular gut microbe. And if you transfer this gut microbe into the old animals, how does they behave? So for this, what we did initially, we took animals, and from literature, we identified that people who they use, young animals, usually they are 2 months old, and the animals which are old, they are 22 to 24 months old. So same way we selected such animals, and these animals, young animals, we treated with antibiotics so that we induced dysbiosis. So initially what we did, we took bone marrow cells from these animals, and then we developed them into dendritic cells with the help of IL-4 and GM-CSF. And then we analyzed these dendritic cells. At the same time, we also analyzed the gut microbiota of these animals, and we observed that there was a substantial decline in both aerobic and anaerobic population of gut microbes in old as well as the young, which the gut was disrupted through treatment through antibiotics. So it was quite amazing to see that the aerobic, both anaerobic and aerobic bacteria, they decreased significantly. But interesting part was that when we looked into the cell number of the bone marrow cell number, we observed that there was no difference upon dysbiosis or old animals. However, these bone marrow cells, when we use them to transform into dendritic cells, so we observed there was a substantial decrease in the capacity of these bone marrow cells to get differentiated into dendritic cells, both in old as well as dysbiotic animals. So next, we thought that these dendritic cells, whether they have lost their phenotypic capacity or the functional capacity, or is there any change in the function as well. So then we monitored the important molecules, that is CD886, MSC class 2 molecules. Since dendritic cells, as I told you, they are important cells in activation of naive T cells. And to activate naive T cells, they should express optimum level of MSC molecule as well as co-stimulatory molecules. So interestingly, what we observed that these animals, which the old animals, they were expressing high level. There was upregulation in the expression of CD8, T86, and MSC class 2 molecules, indicating they acquire the hyperactivation activity. So next, we also looked into the expression of CD80 molecules, and we also observed that there was increase in CD40 as well. So CD40, CD80, 86, they are considered to be co-stimulatory molecules, and these molecules are important for the optimum activation of T cells. So TIM3 is one of the molecules which is responsible for immunosuppression. So surprisingly, what we observed that these cells, dendritic cells, they showed downregulation in the expression of TIM3, and this we observed both through flow cytometry and as well RT-PCR. Now the important thing was whether these dendritic cells have capacity to phagocytose antigen, whether they phagocytose antigen, they can they uptake antigen, and then whether they can process and present this antigen to the T cells. So whereas in young animals, we observed through flow cytometry that they could uptake substantially the antigen, and whereas the dysbiotic animals, they showed downregulation in the uptake of the antigen. And this we showed through flow cytometry as well as we showed through confocal microscopy. So you see here that in young animals, there was sufficient uptake of dextrin FITC, whereas in dysbiotic or old animals, they could not uptake much of the antigens, indicating that the dendritic cells from old animals, they also lose their capacity for antigen uptake. So next was whether they have a capacity to activate T cells. So to check the functionality of dendritic cells, we took both syngeneic CD4 T cells as well as allogenic T cells. And these T cells, they were anti-CD3, CD28 activated. And then we cultured them with dendritic cells so that we provide equal physiological conditions to young as well as old T cells. So in that case, we observed that young animals, they optimally activated T cells, whereas the old and dysbiotic animals, they showed hyperactivation of these T cells. So these T cells, these dendritic cells, they could even activate the naive T cells as hyperactivation of naive T cell, and also they could break down tolerance. So the important part was that what exactly is responsible in these dendritic cells so that they are hyperactivating these T cells and breaking down tolerance of tolerized T cells. And we observed that the secretory molecules, especially IL-6, IL-1 beta, TNF-alpha, IL-12, interferon gamma, these are all secretory molecules, pro-inflammatory cytokines, they were highly upregulated as compared to young animals when they were compared with old animals or young dysbiotic animals. So the young animals, if you treat them continuously with antibiotics, their guts start behaving like old animals. And these bacteria, some of the bacteria, since they disappear, ultimately leads to generation of more of hyperactive dendritic cells rather than and decreasing the frequency of tolerogenic dendritic cells. So it was quite interesting and intriguing to note that the anti-inflammatory cytokines like IL-10, IL-2, TGF-beta, IL-4, they were highly down-regulated. So these cells, the dendritic cells, they started producing less amount of tolerance or of immunosuppressive cytokines, indicating the majority of these dendritic cells, they are of activation phenotype rather than tolerogenic dendritic cells. So besides these, using ELISA, we also confirmed our result through RT-PCR, and we could reproduce these results with RT-PCR as well. So all pro-inflammatory cytokines, they were increasing, whereas anti-inflammatory cytokines, they were decreasing. So further, we wanted to see whether these genes which are responsible in controlling or suppressing the secretion of activation molecules, and they are responsible of suppressing, or they are highly upregulated in tolerogenic dendritic cells. What is the status of those molecules? So then we start looking into IRF4, IRF8, PDL1, BTLF4, and ALDH2. Interesting part was that all these molecules, we found that they were highly downregulated in old and young dysbiotic animals compared to the young one. So these molecules are already established to play a suppressive role in dendritic cells. So this indicates that these dendritic cells, they are of high activation profile, and they shift towards more of activation profile rather than tolerogenic dendritic cells. So then we thought to look into the phenotype of T cells, especially whether it can, what is the status of regulatory T cells, since regulatory T cells are quite important in suppressing the immune system. So again, we observed that the old animals, they showed very low percentage of regulatory T cells as evidenced by the expression of FOXP3 and CD25, as well as there was high expression or high percentage of regulatory T cell in young as compared to the old. So we also, now we started looking into the mechanism. What exactly is the mechanism which gene is ultimately controlling the hyperactivation of these dendritic cells in old animals? So one of the key molecule is responsible for homeostatic signaling is NF-kappa B. So NF-kappa B, it drives the expansion of the activated cells, and these tolerogenic dendritic cells, they have a lot less of NF-kappa B. So next is that we started looking on this activation marker, NF-kappa B. So we looked into its phosphorylation of P65. So we observed that in young animals, there was very less expression of NF-kappa B, whereas there was a high expression of NF-kappa B in both young and dysbiosis. And so we also observe this through confocal microscopy by staining. It's one of the subunit P65. And if you see here that this subunit was highly expressed in dysbiotic and old animals, but not in young animals. So we also looked into these other immunosuppressive markers where they are considered to play a crucial role in case of dendritic cells like IDO and iNOS, nitric oxide, arginase. And then in this case, we observed that at the gene level, there was a downregulation in express of iNOS, which is considered to be important in providing tolerogenic capacity to dendritic cells. And then we also observed in downregulation in nitric NO and then arginase as well as IDO. So these molecules, they play a very crucial role in regulating the tolerogenic function of dendritic cells. So this, besides through RT-PCR, we also showed it through Western blot just to substantiate our data. So similar results we observed with the Western blot as well. So now, the important thing what we wanted to see is that what exactly is going on in the gut? How is the gut modulated? So we did, looked into through NSG, we looked into relative abundance of the phylum, and we observed there was a downregulation in firmicutes, and whereas there was increase in the level of bacteroids. So these bacteroids, they are considered to be important for the activation of, for activating the function of dendritic cells. So in this case, we observed that in old animals, there was high level of bacteroids, whereas less level of firmicutes. And then we looked into abundance of genus, and it was lactobacillus, which ultimately we observed. And then we looked into the species level, and it was found to be relative abundance of lactobacillus plantarum. So if you see here, in this case, as compared to young, there was a substantial decrease in the level of lactobacillus. So this lactobacillus, ultimately, we started looking into these. We took this lactobacillus and it was identified to be lactobacillus plantarum. So this lactobacillus plantarum we used to gavage these animals, and then we looked into whether this gavaging can revert back the functional activity of dendritic cells. So we gave this lactobacillus plantarum into the animals which were old, and then we compared the relative abundance in their gut of lactobacillus plantarum. So we monitored for 3 different time points, and we observed that the animals which were not given lactobacillus plantarum, the old animal didn't show any change in the frequency, whereas when lactobacillus plantarum was given to these animals, there was substantial increase in the frequency of lactobacillus plantarum in the gut of these animals. So the next is since we observed earlier that with age there is the breakdown of epithelial barrier in the intestine. So when these old animals, we provided lactobacillus plantarum, and then through Dexton FITC, we observed whether it goes into the blood. So we measured this lactobacillus, we measured Dexton FITC in the blood, and we could not see much in the blood. So indicating that the permeabilization or epithelial barrier, it becomes intact upon given lactobacillus plantarum. So they start behaving like gut of young animals. So next is, then again, we started repeating same experiments as we observed in case of old animals. We gave lactobacillus plantarum, and when we observe the expression of CD86, we observed CD8086, and then we observe that the expression of this 86 or 80, it was optimally expressed compared to young animals. So there wasn't heightened expression of this molecule, and same way we observed the expression of CD40. You see this CD40 was also not highly expressed, it was optimally expressed as seen in young animals. And same way we observed that this TIM3, which was down-regulated, it started coming up-regulated in case of old animals. So we next wanted to see the phagocytic activity of these dendritic cells. So they retained phagocytic activity upon giving Lactobacillus plantarum. And this was shown through flow cytometry as well as through confocal microscopy. So if you see here that upon feeding Lactobacillus plantarum, the old animals, they showed equally good phagocytic activity like of young animals. So old showed substantial decrease in phagocytic activity. So indicating that upon transfusion of Lactobacillus plantarum, the animals, they start acquiring the young phenotype in their gut. So same way we observed that the hyperactivation molecule of pro-inflammatory cytokines, they were down-regulated. All of these cytokines, whereas anti-inflammatory cytokines, IL-10, TGF-beta, IL-2, IL-4, they increased as compared to the old animals. So there was an increase in these cytokines. So indicating that these dendritic cells, which they started acquiring more of tolerogenic phenotype. And this was further substantiated the result through RT-PCR, the gene level, and we observed that all these pro-inflammatory cytokines, they start decreasing, whereas anti-inflammatory cytokines, they started increasing. So we also looked into the genes which are directly responsible in case of dendritic cells for inducing tolerance. So like RF4 is increased, RF8 increased, PD1 started, PDL1, PDL2, and BTLA4 and ALDH2, all of them, these molecules, they start increasing. So these dendritic cells start acquiring a normal phenotype, a phenotype which is responsible not to break down tolerance, not to induce autoimmunity, rather than inducing optimal activation of T cells. And same way we observed other tolerogenic molecules like nitric oxide, inos, and these molecules, they are directly responsible for the activity of dendritic cells. IDO is considered to be the best molecule responsible for induction of tolerogenic phenotype in dendritic cells. So it was increased, and this was shown through RT-PCR as well as through Western blot. And you see over here, whereas the inos and IDO, they were highly down-regulated in case of old, upon Lactobacillus plantarum, that was upregulated. So besides IDO, this arginase was also upregulated. So replenishment of the gut microbes with the Lactobacillus plantarum, it also revived the metabolic shift in these animals, as we observe the expansion through the population of NBD positive bone marrow derived cells. So this was highly increased in NBDG uptake, and this could also be observed through confocal microscopy. So then it was important to see that what is the function, what is the activity or the impact on NF-kappa B, NF-kappa beta, since it was modulating, it was the main molecule responsible for the hyperactivation of dendritic cells. And upon Lactobacillus plantarum, we observed that there was substantial decrease in the expression of the NF-kappa B, plus its other molecules like REL1A And also subunit of NF-kappa BP365 we observe through flow cytometry as well as through Western blot. And you can see here that there was substantial decrease, and this was further reproduced by confocal microscopy. And here we could observe that there was substantial decrease upon Lactobacillus plantarum given to old animals compared to the old animals. So you can see visually that these old animals' dendritic cells are expressing high level of NF-kappa B compared to old animals were given Lactobacillus plantarum. So now the important part is upon the functionality of these T cells. So whereas these T cells were responsible for hyperactivation, and when we use same system like previous one, we cultured these dendritic cells with syngenic CD4 T cells and allogenic CD4 T cells. And then we observed that like old cells, they were inducing hyperactivation of syngenic as well as allogenic cells. So upon Lactobacillus plantarum, we observed that there was a downregulation in the proliferation of CD4 T cells. And most of these cells which we observe, these were FOXP3 positive. So you can see here, these were gated on CD4 positive, CD25 and FOXP3 cells, syngenic cells. So as compared to old, there was a statistically significant increase in the level of FOXP3 cells and as well as in allogenic cells as well. So then we wanted to see whether these dendritic cells, since previously they showed downregulation in the migratory markers, especially CCR7 and CCR5, these two molecules are CCR9. These two molecules, chemokine receptors, they are considered to be very crucial in helping migration of dendritic cells. Since these dendritic cells, they had lost their capacity of migration since they were downregulated in old animals. So when Lactobacillus plantarum was given, we observed that there was remarkably there was increase in the expression of these molecules. So both CCR7 and CCR9, they were highly upregulated. So indicating that these animals, they show these young animals, they start expressing high level of activity in dendritic cells. Same way we observed in the expression of FOXP3, that is in old animals which were given Lactobacillus plantarum, we observed that they acquired activity. And then we did transfer experiments from old animals treated with Lactobacillus plantarum. We transferred these regulatory T cell into young animals, and we observed that these animals, they start behaving like normal animals. And then we looked into the gene through gene expression microarray. We
Hello?
did RNA.
Javed,
Yeah, just
sorry
done.
Sorry to interrupt.
Just
If
done.
you can wind up, yeah, sure.
Yeah,
Thank you.
done. So we looked into metabolic genes, we looked into cell cycle DNA expression. So looking into the mechanistic illustration of the studies ultimately indicate that with age, there's a disruption in gut microbiota. And this disruption in gut microbiota ultimately it influences the function of dendritic cells. And then these dendritic cells, they acquire hyperactivation property. And because of this hyperactivation property, they have capacity to even break down the tolerance of T cells, which are undergone central tolerance in the peripheral blood. So this is why we get a lot of autoimmune diseases with old age and other inflammatory diseases. So I'm going to stop here, and I'll be happy to take any questions.
Thank you so much, Javed, for the exciting talk and sharing the animal data linking aging with the gut micro composition and also immune dysfunction. And you have come up with some therapeutic intervention in the form of a species of Lactobacillus plantarum. It's a very good lead. We'll take a few questions. please.
Wonderful talk, sir. So how did you home in on only one species? There might be many others in that particular phylum that you showed. What was the criteria for that?
So the criteria was, although there was upregulation, but what we observed that in Lactobacillus plantarum, it was the maximum decline in the gut of old we observed was Lactobacillus plantarum. And that is the reason ultimately we decided on this.
Out of all the candidates that were there.
Above all the candidates. Yes.
There is one question from here.
This side, chairperson.
Sir, probiotics with anti-aging ability refers as zerobiotics. I hope I'm.
Can you speak loudly?
Probiotics with a delay in aging, we refer them as a zerobiotics.
Zerobiotics.
Zerobiotics. So with reference to the zerobiotics, because you took only one culture, why not checking multiple strains and looking which is the best one with reference to the gut microbiota modulation as well as immunomodulation and dendritic function also.
See, even though there were a lot of dysregulation in other species as well, so we have chosen over here. Initially, we started using different strains.
Okay.
But what we observed in that, if you take a consortium of bacteria, so they were, reciprocally regulating each other. So you mix particular proportion, and when we injected them, we could not see their retention in gut. Plus, the frequency was frequently changing. So that was the reason we tried to stick to one particular species. And since one Lactobacillus plantarum was giving us a substantial difference, and that was the reason we selected this for population.
Thank you.
Actually, this question crops up again and again. So since you were ultimately targeting the immunological cells and that two dendritic cells which use process antigens, like Bhavtosh once did the complete NGS of Lactobacillus rhamnosus, and there were 23 different types after the full sequencing. And they have motifs which through the different bioinformatic tool, one could find their functions. So picking up antigens or cocktail of antigens to trigger could be one of the other bases to find out. And a lot of these signatures are now available in the literature for most of the probiotic strains. But one of the things which I found very interesting over here, that they could actually stimulate the exhausted T cells. So in the CAR T cell therapy, which are coming up these days, after some time the exhaustion of T cells happen. And there are a lot of strategies which are being put up. Different kind of compounds are given to, again, bring back those T cells from the exhaustion. So this could be a very good approach to move them. But I don't know whether in those sterile conditions, these could be given as a whole organism. But maybe an approach to look at the antigens or subunits could be also one approach.
Yeah, this is a very interesting question. And definitely, if in such cases, like especially you're talking in context with cancer, instead of giving whole bacteria, if we identify some of the metabolites, so that will be quite interesting. And giving Lactobacillus plantarum, we could observe that there was the cells which underwent exhaustion or the cells which were tolerized. When we cultured with these cells, dendritic cells which were stimulated, we obtained from animals which we injected Lactobacillus plantarum. So they had a capacity to induce the proliferation of tolerized cells or exhausted cells.
So
There's one question from here. Last question, please.
my question is the same extension of what Professor Ganguly has asked. So we are doing CAR T cell therapy and as well as bispecific T cell therapy or T cell engaging therapy. And one of the problems which we face is the T cell exhaustion which happens in these patients, and the CAR T cell therapy or the bispecific therapy that doesn't work. So one of the things which is coming up is the use of short chain fatty acids, the diet which is rich in short chain fatty acids. And so, I know you have worked on Lactobacillus plantarum, but do you think, what is your view on, use of the short chain fatty acid diet, the diet which is rich in this?
Yeah, again, this is a very interesting question. So we are now looking into metabolites. So whether it's short chain fatty acids or some other metabolites, which are important for overcoming exhaustion in T cells. So that will be interesting if you feed these short chain fatty acids or some other metabolites, whether in such cases you can break down tolerance or tolerized cells or exhausted cells. So this has quite potential because in many cases, especially in immunosuppression, even lactobacillus, they start proliferating to an extent that it becomes difficult to control. And another problem we faced was the colonization of lactobacillus in the intestine. So for that was the reason that we have to give regularly. You might have noticed in our experiment design that we give regularly, constantly we give for many days Lactobacillus plantarum. So in that case, even the lactobacillus or any antigen you keep on exposing the immune system, it becomes foreign. So even the tolerized cells, they may get activated. So giving the metabolites may be a very ideal situation. It may be a very judicious approach.
Thank you.